Welcome to Biomania Revision Podcast.
Hello, everyone, and welcome. It's great to be back for another super detailed and hopefully energetic deep dive. I really hope your prep is going well and that you're starting to really enjoy these concepts.
Yeah, today's session is going to be a little different. Usually with biology, we get stuck trying to just, memorize things, rote learn diagrams. But today we're going to decode a chapter that is actually all about logic, mathematics, and even a bit of chemical engineering. Our target today is Chapter 13, Concepts, Plant Growth and Development. I've got the textbook here, some really detailed handwritten notes focusing on growth rates and hormones, and frankly, a ton of questions. Our mission is simple, to understand how plants actually grow.
Right.
Not just, well, the tree got taller, but what's the magic happening at the cellular level?
Absolutely. And this chapter is so interesting because it perfectly bridges that gap between anatomy and physiology. It's not just important for your exams, but it's crucial for understanding how this entire kingdom of organisms actually functions. We're going to look at how a tiny seed pushes up against gravity, how it decides when to flower, and how these chemicals, these hormones, conduct the entire orchestra.
So basically, today is all about the how and the why. I'm going to be the student asking all those questions we usually skip over or get confused by, and you're going to walk us through the mechanics of it all. So are you ready to crack the code of plant life?
I am always ready. Let's dive in.
Okay, let's unpack this. First things first, let's start with the absolute basics. We use the word growth so casually. My hair grew, I grew taller, even mountains grow because of tectonic plates. But for an exam, biologically, what is the exact definition of growth?
That's a great place to start because the biological definition is very precise. Growth is defined as an irreversible, permanent increase in the size of an organ or its parts, or even of an individual cell.
Okay, so there are two key words there that jump out: irreversible and permanent.
Exactly. Those are the ones you have to remember.
So irreversible means it can't go back to its original size.
Precisely. Once a leaf expands, it can't just shrink back down. It's a permanent change. But let me ask you a question, one that's highlighted in these notes and trips up a lot of students. If we take a piece of dry wood and soak it in water, it swells up, right?
Right, yeah.
The size increases, the volume increases. Would we call that growth?
Technically, the size did increase.
Yes.
So it seems logical to say yes, it should be growth.
And that's the trap. It is not growth. The notes specifically call this process imbibition. It's a physical process where solid particles absorb water. But the most important thing is it's reversible.
Ah, okay.
If you put that piece of wood back out in the sun, the water will evaporate and it will shrink again. True biological growth, by definition, cannot go backwards. It almost always involves metabolic processes, both anabolic and catabolic, and the synthesis of new material.
Got it. So if it's reversible, it's not growth. Only permanent expansion counts. But if we go even deeper, what does growth mean at the cellular level? What is actually increasing inside the cell?
That's the real question. At the cellular level, true growth means an increase in the amount of protoplasm. The protoplasm is all the living content of the cell. The cytoplasm, the nucleus, the organelles. It's only when the quantity of protoplasm increases that a cell can divide or elongate. Without more protoplasm, growth is impossible.
But protoplasm is sealed inside a cell wall. You can't just take it out and weigh it on a scale. I can't imagine saying, oh, this cell's protoplasm increased by 2 micrograms. It sounds impossible to measure easily in a lab without killing the cell.
You're exactly right. Measuring protoplasm directly is incredibly difficult. So scientists came up with some proxies or parameters that represent growth. We measure it indirectly.
Okay, so what are they?
Things like fresh weight, where you just weigh the plant part, or dry weight, which is the weight after you've removed all the water.
Which one is more accurate?
Oh, dry weight is considered much more accurate because the water content in fresh weight can fluctuate wildly depending on the environment.
Okay, makes sense. What else?
We can also use length, for height, area, which is especially useful for leaves, volume for things like fruits, and of course, cell number, how many new cells were made.
Cell number. You mean we actually measure growth by counting cells?
We can, yes. And this is where we get to some mind-blowing stats that are in the notes. Numbers in biology can sometimes feel a bit dry, but these ones are just insane.
Hit me with them.
Example number 1, the root apical meristem of a maize plant. A single root tip meristem can produce, per hour, 17,500 new cells.
Wait, hold on. 17,500 cells every hour. from just one root tip.
Yes, it's like a biological machine gun just firing out new cells.
Yeah.
So here we're measuring growth in terms of the increase in cell number.
That's incredible.
And then example number 2, a watermelon. Here, it's less about the number of cells and more about the size of the cells. A single cell in a watermelon can increase in size by up to 350,000 times.
Whoa, 350,000 times larger.
It's massive. So in this case, growth is measured as an increase in size of the cell. Growth isn't just about numbers, it's also about volume.
So the parameter you use really depends on the organ you're looking at.
Correct. If a pollen tube is growing, we measure its length. If a dorsiventral leaf, like a typical dicot leaf, is expanding, we measure the increase in its surface area.
Speaking of length, the notes mention an instrument called an axonometer. What is that?
It's also called an oxenometer, often an arc oxenometer. It's a simple mechanical instrument, really, that's often used in school labs to demonstrate linear growth.
How does it work?
It has a pulley and a pointer system. You tie one end of a thread to the plant's stem and the other to a weight. As the plant grows taller, it pulls the thread, the pulley turns, and the pointer moves across a scale. It basically allows you to see and quantify growth in real time. Think of it as a speedometer for plant growth.
Got it. Okay, so growth is measurable. Now let's move to a fundamental difference that always used to confuse me until I saw this comparison chart in the notes. Plant versus animal growth. The notes say plants have open growth and animals have closed growth or determinate growth. What's the deal with that?
Well, think about animals. Our growth is determinate. How long did you grow taller for? Maybe until you were 18, 20?
Yeah. And there's a hard stop. I can't just decide to get taller now.
Exactly. And the number of our organs is fixed. We have 2 kidneys, 1 liver. We don't sprout new organs later in life. Our growth plan is closed. But plants, their growth is indeterminate or open.
Open.
Open means that the plant retains the capacity to add new cells and new organs for its entire life. A 100-year-old banyan tree will still produce new branches, new leaves, new roots. It never really gets old and stops growing in the same way we do.
And what gives them the superpower? Why can't we do that?
They get this power from specialized regions called meristems. Meristems are areas where cells are in a state of continuous division. They never fully mature. They remain in an embryonic state. Basically, plants have permanent pockets of stem cells.
Right. And there are different types.
We mainly classify them by location. First, you have the apical meristems, which are at the tips, the apex of roots and shoots. Their job is to increase the plant's length or height. We call this primary growth.
And the other type.
The others are lateral meristems, like the vascular cambium and cork cambium. These are responsible for increasing the plant's thickness or girth, and that's called secondary growth. The notes clearly point out that monocots like grass or corn usually don't have this. That's why a blade of grass gets long, but its stem doesn't become thick and woody.
So plants never stop growing because they have this magic factory called a meristem. That's really cool.
It is. Meristems truly are the engines of plant life.
Okay, so we've defined growth. Now let's zoom into the microscopic level. The notes have this diagram of a root tip, and it's marked with 3 distinct zones. Let's trace the life of a single cell. If I'm a brand new cell, just born at the root tip, what's my life journey going to look like?
Great visualization. Let's start right at the tip of the root and move upwards, or proximally. Your journey will take place in 3 phases of growth. Imagine you're standing in a queue.
Okay, I'm in line.
Right at the front of the line is phase 1, the meristematic phase. You're at the root apex. You have one job and one job only: divide. Constant, rapid division.
What are my characteristics?
Well, your protoplasm is very rich and dense. Your nucleus is large and conspicuous, easy to see. Your cell walls are primary walls. They're thin, made of cellulose, and very flexible. And for communication, you have tons of plasmodesmatal connections with your neighbors.
So I'm just a hyperactive dividing cell.
And according to the notes, your respiration rate is extremely high because all that division requires a massive amount of energy.
Okay, so I've divided. The new cells behind me have pushed me forward, so I'm a little bit away from the tip now. What happens next?
Now you enter phase 2, the elongation phase. This zone is just proximal to or behind the meristematic zone. Here, division stops. The goal now is to get big.
Yep.
3 major changes will happen to you, and these are often asked about in sequence on exams. First, increased vacuolation. Before you had tiny vacuoles or none at all. Now large vacuoles will start to form and fill with water.
Why is that so important?
Plant cells mostly grow by filling vacuoles with water. It's a kind of cheap growth. You get a huge increase in size with minimal investment in new material, all thanks to water turgidity. Second, cell enlargement. Your size will increase dramatically. And third, new cell wall deposition. New wall material will be laid down on the inside of your thin primary wall to support your new, larger size.
So this is where I get tall. And after that?
After that comes phase 3, the maturation phase. This is even further behind the elongation zone. Here, you've reached your maximum size. Now it's time for differentiation.
Time to decide what I want to be when I grow up.
Exactly. You decide on a career. Your cell wall will finish thickening, and your protoplasm will be modified to perform a specific function. If you're destined to become xylem, you'll actually die and become a hollow pipe for water. If you need to be a root hair, your wall will extend out into a long thread.
So it's a simple roadmap, really. Meristematic is like childhood, all about division. Elongation is like the teenage growth spurt. And maturation is adulthood, getting a job, specializing.
That is a perfect analogy, and that sequence is always maintained in any growing root or shoot tip.
All right, let's switch gears a bit to the math. Biology students usually run from math, but here in growth rates, we've got formulas. The notes mention two terms, arithmetic and geometric growth. What's the logic here?
Don't worry, the logic is very simple. Growth rate just means the increased growth per unit of time. Let's start with arithmetic growth.
Okay.
Imagine mitosis. One cell divides to make two. In arithmetic growth, of those two daughter cells, only one continues to divide. The other one differentiates and matures. It drops out of the race.
So you're only adding one dividing cell at each step.
Exactly. So you go from one dividing cell to 2 cells total where one divides and one stops, then 3 cells total where one divides and 2 have stopped, then 4, and so on. If you plot this on a graph of length versus time, you get a linear curve, a straight line.
And the formula?
The formula is light dollars plus RT, where light dollar is the length at time t, light dollars is the initial length at time 0, and R is the growth rate, which is just the slope of that straight line.
Okay, so that's slow and steady, constant growth, like climbing a ladder one step at a time. What about geometric growth?
Geometric growth is explosive. Here, after division, both daughter cells retain the ability to divide.
So it doubles each time.
Yes. 1 becomes 2, 2 become 4, 4 become 8, 16, 32. It's exponential. This is common in things like early embryonic development when a zygote is becoming an embryo. Its graph isn't a straight line, it's a sigmoid curve, or an S-curve.
The S-curve, that's the one you see everywhere in nature, isn't it?
It is, because in the real world, no living organism can grow exponentially forever. Resources always become limited. The S-curve has 3 distinct phases.
Right.
First is the lag phase. Growth is slow at the beginning, the cells are just adapting, getting the machinery set up. Then comes the log phase, or the exponential phase. This is where growth just skyrockets. Resources are abundant, and the population is increasing at maximum speed.
And then it has to slow down.
Right. It enters the stationary phase. As nutrients start to run out or space becomes limited, growth slows down and eventually stops. The curve flattens out. The formula for this is 1 over $, base W dollar A door. It is the base of the natural logarithm.
Now, in that formula, the notes have a specific term for R. It's called the efficiency index. What's that about?
Good catch. In this context, R is the relative growth rate. It's called the efficiency index because it tells you how efficiently a plant is producing new material relative to its initial size.
Okay.
Do you remember the 2 leaves example from the textbook? The diagram with leaf A and leaf B?
Yes, I do. Leaf A is small and leaf B is much bigger to start with. Let's break that down for everyone listening.
Let's do it. So leaf A has an initial area of 5 centimeter introduce. Leaf B has an initial area of 50 centimeters. After 1 day, they both grow by an additional 5 centimeters.
Okay, so leaf A goes from 5 to 10 centimeters.
And leaf B goes from 50 to 55 centimeters torts. Now, their absolute growth is identical. Both added 5 centimeters of material.
Right.
But what about their relative growth? Leaf A went from 5 to 10. It literally doubled in size. That's a 100% increase.
And leaf B only went from 50 to 55. That's, what, a 10% increase?
Exactly. So even though their absolute growth was the same, leaf A R, its relative growth rate or efficiency index, was 10 times higher.
That makes perfect sense. It's like a startup versus a massive corporation. The startup, leaf A, doubles its size, while the corporation, leaf B, barely grows in percentage terms, even if the raw number added is the same. Efficiency really matters.
Absolutely. The relative growth rate gives you the true picture of biological efficiency.
All right, now let's tackle some of those biology terms that feel like tongue twisters. Differentiation, de-differentiation, re-differentiation. It sounds like a rap lyric. What is the actual difference between them?
It's actually very simple if we just walk through it step by step. We already know differentiation, that's a cell taking on a job. A meristematic cell stops dividing and modifies itself for a specific function.
Right.
The classic example is a tertiary element, like a xylem vessel. Its job is to transport water, so it gets rid of its protoplasm to become a hollow tube, and it reinforces its walls with a tough, strong lignocellulosic material so it doesn't collapse under the high tension of the water. That's differentiation.
Okay, the cell has settled down in this career. So what is dedifferentiation?
Sometimes a plant has an emergency and needs new cells in a place where cells have already matured. So cells that were differentiated and had stopped dividing, like parenchyma cells, can actually regain the capacity to divide.
They go back to being meristematic.
Essentially, yes. This back-to-school moment is called dedifferentiation. The best example is the formation of the intervesicular cambium and the cork cambium. These tissues are needed for secondary growth, and they form from mature, living parenchyma cells that reactivate.
So it's like retired workers being called back to the workforce for a special project.
That's a great way to put it. And that brings us to redifferentiation. The cells that were formed by that dedifferentiated cambium, they then divide for a while, and then they lose the ability to divide again as they mature into their new final function.
So they specialize a second time.
Correct. So the cells of the secondary cortex, or secondary phloem, are redifferentiated. The cycle is divide, mature, differentiation, regain division, dedifferentiation, divide, and then mature again, redifferentiation. Nature is incredibly good at recycling talent.
Wow. And through this whole process, the plant has another ability, right? The notes call it plasticity.
Yes, plasticity.
Does that just mean being flexible, like plastic?
You've got it. Plasticity is flexibility. It's the ability of plants to follow different pathways and form different kinds of structures in response to the environment or different phases of life. An animal's body plan is usually quite rigid, but plants can adapt and change.
And the best example of this is heterophyly.
The perfect example. Hetero means different. Phyly refers to leaves, so having different types of leaves on the same plant. And the examples for this are very specific and come up on exams directly.
So what are the key examples?
Well, there are two main types of heterophyly. The first is developmental heterophyly. This is where the leaf shape changes with age.
Okay.
The classic examples are cotton, coriander, and larkspur. If you've ever seen a coriander plant, you'll know that the leaves near the bottom, the juvenile leaves, are broad, while the leaves higher up on the mature plant are thin and feathery.
I have noticed that.
Right.
And the second type?
The second is environmental heterophyly. This is where the environment dictates the leaf shape. The textbook example is the buttercup, or ranunculus. The leaves that are submerged underwater are highly dissected and ribbon-like,
Why is that?
so that the water current can flow through them without tearing them apart. But the leaves on the same plant that are above the water in the air are broad and lobed to maximize sunlight capture for photosynthesis. Same plant, same genetics, but the environment determines the final form. That is plasticity.
That is an amazing adaptation. Plants are genuinely smart. So we've covered growth, differentiation, plasticity.
Yeah.
But who is controlling all of this? How does a cell know, okay, time to divide or time to ripen or now I need to close my stomata?
Yes?
This must be where our chemical messengers come in, the plant growth regulators, PGRs.
Yes, the PGRs. We also call them phytohormones. They are small, simple, organic molecules, but their impact is absolutely massive. You only need them in tiny quantities, but their effect is felt throughout the entire plant.
And we can group them right.
Broadly, yes. We divide them into two main groups. First, the plant growth promoters. These are the ones that encourage growth activities like cell division, cell enlargement, flowering, and fruiting. This group includes our three heroes: auxins, gibberellins, and cytokinins,
And the second group.
the plant growth inhibitors. Their role is often related to responding to stress or wounds and inducing dormancy. The main one here is abscisic acid, or ABA.
And where does ethylene fit in all this?
Ethylene is the interesting one. It can fit into both groups, but it's largely considered an inhibitor for many of its functions, like senescence and abscission, even though it also promotes fruit ripening. Most textbooks place it alongside the inhibitors.
Okay, let's do a deep dive on each of these big five hormones. Their discovery stories, their functions, and how we use them in agriculture. I feel like every hormone has its own unique story. Let's start with auxins. Can we call this one the grower, or maybe the boss?
The boss is very fitting. The discovery of auxin is fascinating. The credit really goes to Charles Darwin and his son, Francis Darwin. They observed that the coleoptiles of canary grass, that's the protective sheath covering a young shoot, would bend towards a light source.
Phototropism.
Right, phototropism. And Darwin concluded that some influence was transmitted from the tip of the coleoptile downwards, causing the bending. Later, a scientist named F.W. Wendt actually isolated this chemical from the tips of oat seedlings and gave it the name auxin, from the Greek word auxin, which means to grow.
And what's the main natural auxin in plants?
The primary natural auxin is IAA, or indole-3 acetic acid. There's also IBA. These are mainly produced in the growing tips of shoots and roots.
Oh.
And then there are synthetic ones that we've made, like NAA and the famous 2004D.
Okay, so what are its main functions, the ones we can actually see? I know apical dominance is a huge one.
Apical dominance is probably auxin's most prominent effect. The auxin produced in the apical bud, the very tip of the shoot, actually inhibits the growth of the lateral buds, the ones on the side.
So the plant just keeps growing straight up, but not out to the sides, not butchy.
Exactly. The auxin is basically saying, I'm the boss here, only I get to grow.
So what if you want a bushy plant? Like a gardener would?
Then you cut off the tip. This is called decapitation. As soon as you remove the tip, you've removed the source of the auxin. The inhibitory effect is gone, and the lateral buds are free to grow. The plant becomes bushy.
Ah, so that's what's happening in tea plantations and with hedge making.
That's precisely the technique. The gardeners are constantly trimming the tops to encourage lateral growth, which means more leaves and a denser plant.
Mind blown. That's why trimming is so important. What else does auxin do?
Well, it promotes rooting in stem cuttings. If you're trying to propagate a rose from a cutting, dipping it in auxin powder helps it grow roots faster. It can promote flowering, for example, in pineapples.
And abscission, the dropping of leaves and fruit.
This one's interesting. Auxin actually prevents the drop of young leaves and fruits, but it promotes the drop of older, mature ones. It's part of nature's cleanup crew. It also induces parthenocarpy, the development of fruit without fertilization, giving you seedless fruits, like in tomatoes.
And it's a herbicide.
Yes. The synthetic auxin 2004D is a widely used weed killer. The incredible thing is it selectively kills dicot weeds, the broad-leaved ones, but it doesn't harm mature monocots, like wheat or the grass in your lawn. So farmers can spray their fields to kill the weeds without damaging their crop.
That's incredibly useful. Okay, let's move on to hormone number 2, gibberellins. I'm calling this one the extender because everywhere in the notes it just says increases length.
That is the perfect name for it. And its discovery came from a disease, the bakanai, or foolish seedling disease, in rice in Japan.
A disease?
Farmers noticed some rice seedlings were growing abnormally tall and thin, and then they just fall over without producing any grain. It was discovered this was caused by a fungus, Gibberella fujikuroi. A scientist, E. Kurosawa, found that even sterile filtrates from the fungus could cause the disease in healthy plants.
Meaning the fungus was releasing some kind of chemical.
Precisely. And that chemical was gibberellic acid.
So we got this plant hormone from a fungus, and chemically these are terpenes, right?
Yes. There are over 100 different gibberellins that have been identified, GA1, GA2, GA33, and so on, but GA33 was one of the first to be discovered and is the most studied. And its uses in agriculture are huge. Just as you said, it's an extender.
How so?
Well, in grapes, it increases the length of the stalks, which allows the grapes in the bunch to grow larger and be less compact. In apples, it helps to elongate the fruit and improve its shape, which increases market value.
And sugarcane. The notes have a crazy statistic for sugarcane.
Oh, for sugarcane farmers, this is like gold. Sugarcane stores sugar in its stem. If you spray the crop with gibberellins, the stem length increases dramatically. The textbook says this can increase the yield by as much as 20 tons per acre.
20 tons, just from a spray. That's a massive increase in profit.
It is. It also hastens the maturity period in juvenile conifers, so you can get seed production much earlier. And it promotes something called bolting.
Bolting, like with a nut and bolt.
No. Bolting is the sudden elongation of the internodes, the stem, just before flowering. This happens in plants with a rosette habit, like beets and cabbages, where the leaves are all clustered close to the ground. Spraying with GA makes the stems shoot up and flower.
Got it. Okay, on to cytokinins. The name itself sounds like cytokinesis, so I'm guessing the divider.
Correct, again. Its discovery was also a bit of an accident. F. Skoog and his coworkers were trying to grow tobacco stem callus tissue. They found that the callus would only proliferate if the auxin medium was supplemented with something else, like yeast extract or coconut milk or even DNA. Yes, and they eventually crystallized a substance from autoclaved herring sperm DNA that they named kinetin.
A plant hormone from fish sperm. Biology is so weird sometimes.
It really is. But the important thing to note is that kinetin doesn't occur naturally in plants. The first natural cytokinin that was discovered is called zeatin, which was isolated from corn kernels and coconut milk.
And where are they made in the plant?
Cytokinins are synthesized in regions where there's rapid cell division happening, so root apices, developing shoot buds, young fruits.
So its main job is cell division, but the notes also mention it as an anti-aging hormone.
They do. This is sometimes called the Richmond-Lang effect. Cytokinins delay leaf senescence, that's the aging and yellowing of leaves. They do this by promoting nutrient mobilization. They help direct nutrients to the leaves, keeping them green and functional for longer. This is why they're used commercially to extend the shelf life of fresh produce and cut flowers.
And it has a relationship with auxin, right?
A very important one. It acts as an antagonist to auxin when it comes to apical dominance. While auxin promotes apical dominance, cytokinins help to overcome it, promoting the growth of lateral branches.
So auxin makes the plant tall, cytokinin makes it bushy, a nice balance. All right, next up is ethylene, the ripener. And this one is a gas, isn't it?
Yes, it's the only major PGR that is a gaseous hormone. Its discovery came from an observation by H. Cousins, who confirmed that a volatile substance released from ripened oranges was causing nearby unripe bananas to ripen much faster.
The old one bad apple spoils the bunch saying.
That's literally ethylene at work. It's the most important hormone for triggering ripening. When a fruit ripens, there's a characteristic rise in its rate of respiration called the respiratory climactic. Ethylene is what initiates that process.
What else does it do besides ripening? The notes mention something called the triple response.
Ah, yes. The triple response is seen in seedlings. If you expose a seedling to ethylene, it does three things. 1, horizontal growth, so it grows sideways instead of up. 2, swelling of the axis, so it gets thicker. And 3, the formation of an apical hook in dicot seedlings, which helps protect the delicate shoot tip as it pushes through the soil.
So how do farmers use a gas? It must be hard to spray a gas on a field.
It would be. So they use a compound called ethifon. Ethifon is an aqueous solution that is easily absorbed by the plant, and then it slowly releases ethylene gas inside the plant tissues. It's much easier to apply.
And what do they use it for?
All sorts of things. To hasten fruit ripening in tomatoes and apples so they can be harvested at the same time. For thinning out excess flowers and young fruits in cotton, cherry, and walnut, so the remaining fruits grow larger. In cucumbers, it promotes the formation of female flowers, which increases the yield.
And the deep water rice example is fascinating.
It's a critical adaptation. In deep water rice plants, ethylene promotes rapid internode elongation, which helps keep the upper parts of the leaves and the flowers above the water level so they don't drown. It also helps in breaking seed and bud dormancy, like sprouting potato tubers.
Okay, last but not least, abscisic acid, or ABA. The stress manager.
Right. It was originally discovered under a few different names like inhibitor B and dormin. It's a derivative of carotenoids. And it's widely known as the stress hormone because when a plant is under stress, especially water stress or drought, its ABA levels shoot up.
What does that do?
Its most immediate effect is to stimulate the closure of stomata, which drastically reduces water loss through transpiration. It's the plant's emergency brake to conserve water.
So it switches the plant into a water-saving mode.
Exactly. It's the plant's protector. It also plays a major role in dormancy. It inhibits seed germination. Now, that might sound like a bad thing, but it's absolutely vital. Imagine if a seed germinated right before a long, dry spell. The seedling would die.
So ADA makes sure the seed waits for the right conditions.
It ensures the seed stays dormant, sleeping, until conditions are favorable. In this respect, it's a direct antagonist to gibberellins, which promote seed germination while ABA inhibits it.
So it seems like none of these hormones really work in isolation.
Not at all. That's a crucial point. Plant growth is an incredibly complex process regulated by the interplay of these hormones. Sometimes their effects are synergistic, meaning they work together, like auxin and cytokinin, in promoting cell division.
And sometimes they're antagonistic.
Exactly, working against each other, like ABA and gibberellins in seed dormancy, or auxin and cytokinins in apical dominance. And on top of all this, you have the external factors. Hormones aren't enough. You still need light, temperature, water, oxygen. All of these are essential.
Things like photoperiodism and vernalization.
Precisely. Those responses to light duration and cold treatment are external signals that are ultimately mediated through these internal hormonal signals to control things like flowering.
Phew. That was a massive revision session. We've covered everything from the basic definition of growth to the cells, the math, and the chemicals that run the whole show.
We have. Plant growth isn't just about getting bigger. It's a highly regulated mathematical and chemical symphony. There are checks and balances at every single step.
So what does this all mean for us? I guess the next time you look at a tree or a
Hmm.
flower, you can remember that there's this incredible chemical factory running inside, deciding where the next branch will go and when the next fruit will ripen.
Mm-hmm.
And this is where it gets really interesting, because scientists are now using this knowledge to design crops. We have seedless watermelons, dwarf wheat that doesn't fall over, high-yield sugarcane. And that leads me to my final provocative thought for you to take away.
Okay.
If we can manipulate these hormones to control plant growth so precisely. To all of you listening, that was our deep dive into plant growth and development. Hopefully the next time you look at a tree, you won't just see wood and leaves, but a complex chemical factory that's constantly rewriting the rules of nature. Like and tell how it was. And if you want more chapters to revise like this, then tell in comment section.
Keep learning.
We'll see you in the next deep dive.